Welcome to the Spring 2021 Core Facility Seminar Series. Today we will be featuring the Atomic Force Microscopy Facility, or AFM. I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. In today's seminar, you will hear from Keith Dusoe, the staff scientist working in the AFM Core Facility, as well as working as a postdoctoral fellow in polymer science and engineering. We hope that with these bi-weekly seminars, you will discover what great resources that the centralized UMass core facilities offer to our campus community, the New England region, and beyond. And just a few housekeeping items. The seminar is being recorded, as you heard. If you miss any part of this seminar or would like to forward it to someone who could not attend, there will be a replay of this and all the previous seminars in the series on our website, and I will put a link in the chat. I recommend you set your view mode to speaker. Please stay muted during the talks. We will save the Q&A until the end of the presentation. During that time, I welcome you to unmute yourself and ask your question. And now I'd like to introduce you to Andrew Bernard, the Director of our centralized UMass core facilities. Andrew?
Thank you, Lisa. Welcome, everyone. As Lisa mentioned, my name is Andrew Bernard. I'm the Director of the centralized cores here at UMass Amherst. If this is your first time joining us, we're happy to have you. If you don't know about our cores, we manage state-of-the-art labs, instrumentation, equipment, and world-class experts to help researchers drive their discovery and to pursue scientific endeavors across all disciplines. UMass core facilities are open to anyone from students through senior scientists, regardless of your affiliation, including researchers from other academic institutions and commercial partners as well. To help access our cores, there are several funding opportunities. If you're an IALS investigator, there are core credits and center seed funding available. And for those who may be external to UMass, there's the Mass Innovation Voucher Program, which subsidizes usage for small companies who are based in Massachusetts, as much as 75%. We've already awarded over 500 of these vouchers, totaling more than $5 million in project costs at the UMass cores across all of our campuses. We're here to be your partner and expand your research and productivity. If you'd like access to any of our cores or learn about any specific technologies, please feel free to reach out to me or the individual core directors. Our website has a wealth of information from technology types to information on our directors. Now I'll introduce Dr. Keith Dusoe, who's a staff scientist in both the atomic force and electron microscopy cores. He's going to provide an overview of the possibilities that scanning probe microscopy could add to your research. Look forward to the talk, and I'll be here in the Q&A for any questions. Thank you.
Great. Hello, everybody. Thank you, Andrew, for your introduction. Thanks, everybody, for joining us this afternoon. Again, my name is Keith Dusoe. I'm a staff scientist here at the AFM core facility, in addition to being a postdoc in the Polymer Science and Engineering department, as well as within the Institute of Applied Life Sciences. So just to give you a little bit of idea about myself and to provide an introduction, my background is in material science and engineering. So I have my PhD from University of Connecticut. And my PhD in graduate school work really dealt mostly with small-scale mechanical behavior characterization and some material design and development. And so just to give you maybe just a brief idea of some of the projects I've worked on before, sort of general workflow and thinking about small-scale mechanical behavior, we would be growing single crystalline intermetallic compounds, which the general size of these is pretty small in itself. It's still macro scale, maybe 3 millimeters to 5 millimeters in size, but certainly too small to be used in sort of traditional industrial mechanical characterization equipment. And so what we were doing there was using focused ion beam milling to fabricate and micromachine nanopillars. And so this particular nanopillar is a diameter of 800 nanometers. And it is upon the surface of this parent crystal here. And so after machining this sort of smaller scale geometry, which we would then put it into a nano indenter inside an SEM. And then from there, we can get some maybe impressive movies here. So this is a video of the, it's a miniaturized compression test here. And so this is a diamond flat punch tip. This cylinder here is the nanopillar that was fabricated on our beginning sample here. And then, from this characterization technique, we can get sort of stress strain curves and, really start thinking about the structure property relationships at small scale. So again, just wanted to share with you all where I'm coming from and my background being mostly in small scale property characterization. So going forward to where we are now and where I am now with the Atomic Force Microscopy core, Well, first, I'd just like to really tell you all and share with you all what we have and what we have to offer. And so in our core, we have high resolution instrumentation, right? And in addition to this instrumentation, we also have the capability and the expertise to help with characterization as well as analytical data analysis as well. And so really what we're doing in the AFM core is making this instrumentation available to you all and also supporting whatever your research needs may be. So depending upon where your project is, whether it's an industrial sort of research project or an academic research project, in the AFM core, we're here to help you to go wherever you may be with your particular research project. It could be gathering some preliminary data to maybe support a proposal, right? Some validation of an industrial process if you're in industry, and even we're happy to help you design and start thinking about, well, is AFM right for your characterization needs? And if they are, what is the best experiment? And so almost sort of like a design of an experiment. So that's what we're here for, and we're happy to help you all with that. And just to give you an idea of who currently is using our facility, so we have university users, so those members of the UMass Amherst research communities, as well as the surrounding 5 colleges. And additionally, we have industrial R&D folks using our facilities, and we sort of scan across different technology spaces, right? We have some companies interested in optical fibers, acoustic materials, medical devices. So this is sort of what we're doing here in the AFM facility. And I talked about us having high resolution sort of instrumentation, and so I just wanted to go through and give you just an overview of what instrumentation that we do currently have available, and sort of maybe what you could use this instrumentation for, right? And so we currently have 3 AFMs in our lab, and I'm listing them here, and there's also photos of these here. And so we have the Bruker multi-mode AFM, which is, its strong point is, it's high resolution, right, but also relatively fast scan rate. So I think a scan rate up to about 6 hertz while still maintaining high resolution. We have the Bruker Dimension AFM, which its sort of strong point is a large scanning area. So we can scan at a 100 micron by 100 micron square area roughly. And we also have an Oxford Asylum MFP 3D, which really excels at sort of specialized and customized experiments. And I'll get into that toward the end of the talk as far as what some of the, I guess, possibilities are with when I talk about specialized experiments. But before going there, just to give you an idea of maybe to answer some question of why would I want to use AFM, or when would I want to use AFM, I just wanted to share some data that is currently coming out from some of our users in the AFM facility. And most typically, this sort of falls within the region of surface characterization, right? Things like topography. We have some users who are interested in some lateral force microscopy work as well. And so just to very quickly, just to give you an idea of what AFM is or how AFM maybe works. And so the general principle in AFM is that we have a rectangular cantilever in which a probe is attached to the end of it. And this cantilever probe assembly is then moved across the surface of your sample. And depending upon, a lot of different things can happen, right? Whether your probe is in contact with your sample or not. But essentially what's happening is there's some interaction between the cantilever probe assembly and the surface of your sample. And so this interaction typically is recorded and viewed in terms of a cantilever deflection. And so you can see here, this is an SEM micrograph of just what the typical rectangular cantilever would look like. This is the top side. Underneath it is where our probe would be. And in the AFM, what we're doing is we're measuring cantilever deflection. So this could be torsional or twisting motion, or it could be, I guess, normal from the surface, right? And so as we scan our probe across the sample, we are recording the cantilever deflection, and then from there we can get this sort of 3D image, or I'm sorry, what this is an XY space. At each pixel we have some Z data here. So this is a typical sort of height characterization of a patterned surface here. So 20 by 20 microns square, and this is sort of the general result that we can get out of AFM here. And so there's a couple different modes in which we can operate in, and one of them is something that's called tapping mode. And so in tapping mode, what we're doing is we're taking our cantilever and our probe assembly, and we are driving it or oscillating it at its first fundamental resonance frequency. And so what we're doing is, so we have this shaking cantilever, and we are moving it across the sample surface. And why would you want to use tapping mode? So kind of within the realm of looking at topography, but also in addition to sort of height data and getting an idea of feature size determination. Because we have an oscillating probe, we also have the ability to monitor something called phase, which is whether or not the oscillating cantilever is, if that particular frequency is changing or not, right? And so we could see some change in this oscillation frequency as a result of any sort of chemical interactions between the probe and the sample, as well as any sort of really any other sort of interactive, repulsive, or attractive forces. And so what I'm showing here is from one of our users, Ming Chi, is they have a two-phase system. This is a polymer block copolymer, and you can see here schematically what they're showing is the blue region is one chemical composition, the red region is another, and you can start seeing that. Well, if we look at the height channel, we can see get an idea of topography of our sample, but what's really powerful here is looking at the phase channel, and as we can see that due to the sort of differences of interaction between material 1 and material 2, right, the two different phases in this person's material system, we can sort of get a better idea of, I could say maybe chemical composition, right, some sort of phase distribution of your material within your sample. And so that's one really sort of strong point of using tapping mode, is having this access to not only general topography and height, but also maybe we get some idea about chemical composition and as well here. So going forward, I'd like to talk about another imaging mode that's pretty commonly used and routinely used, and this is called contact mode. And so no surprise here, what we're doing is we're having our AFM probe that's in contact with our surface, right? We have the down force that's applied to our sample fixed, and we are moving across the sample. And so from there, we can get an idea of, again, topography, feature size determination. But also, right, because we're contacting our sample with our probe, we can do things like some mechanical property characterization. So we could do really sort of local probing and get a force displacement curve. You could say maybe per pixel or across the surface of your sample. And additionally, one of our users from the UMass community, Yu Young Jo, they are using LFM or lateral force microscopy. And so these folks are interested in some frictional force characterization here. And so, you can imagine we have our AFM probe in contact with our sample, and we are monitoring the twisting or the torsional force or deflection upon our cantilever. And so from there, we can sort of characterize, or what they're doing is characterizing the frictional force upon their films as a function of the down or normal force. And so this is another example of the type of data that we can acquire and further analyze within our cores here. And so I first started talking about some of the sort of routine sort of characterization efforts we're doing, and I also wanted to talk about some of the more specialized or exotic experiments that we can start thinking about or even support. And so the Oxford Asylum MFP3D is a microscope that really allows us to look at other different operating modes. One interesting mode is something that's called dual AC, or a bimodal tapping mode. So earlier we talked about tapping mode, in which we're driving the cantilever to its fundamental resonance frequency. Well, in this bimodal tapping mode, what we're able to do is to simultaneously oscillate the cantilever at a higher eigenmode or higher a frequency higher than its fundamental. So we have essentially two resonant frequencies that is the cantilever is being driven at here. And so some interesting results can come from here, things like lost tangent imaging. Also, we have capability to look at sort of nanoscale transport measurements. And so we have accessories and holders that allow for conductive AFM to take place. And so I just sort of gave you an idea of what the general resolution or range of current range, 1 picoamp to about 10 nanoamps. And so you start thinking about doing transport measurements and generating sort of force versus IV curves, at small scales, right, in nanoscales here. We also have some other accessories that allow for small molecule characterization. So these are sort of closed fluid cells, which we have really fine control of temperature over. So we can start probing soft materials in a fluid environment ranging from temperatures from ambient temperatures all the way up to 80 degrees C. And also these holders have several ports, so which allows for things like gas or liquid exchange, and also maybe some other probes, or you may want to have in your fluids to allow for monitoring of some other measurement there. And additionally, we can start looking at high temperature for some materials, especially soft materials, right? We can go from 300 degrees Celsius from ambient, and again, we have all these ports for sort of gas exchange and maybe even allow for electrical conductivity, or connection. So really what I wanted to drive home is that in the AFM core here, we can do everything from sort of general surface topography scanning to all the way down to really developing and supporting some experiments that fall within the specialized realm, as I'm showing here. And so in addition to the instrumentation, right, in addition to our support, so myself, I'm there as a staff scientist. Also, our director, Alex Ruby, is very well versed and experienced AFM, and so we are here to support you and your research needs, right? One neat thing we've been doing during this period of COVID-19 is offering remote support during a virtual meeting. And so what I'm trying to show here in this photo is this is a screenshot just from a Zoom meeting, and one thing we've been doing is sharing the AFM computer screen while this is me here on a call with a student or industrial user. And this is pretty nice because it allows for a means for us to remotely collaborate on your research project. So as I'm operating the microscope here, and as we're talking about characterizing your sample, we're able to do that sort of in real time without being in the same lab space together. And so we have remote support available for you all. We also have offering instrument access to individuals right within our local community. So these are students or industrial research communities. Other thing we also offer in our AFM core is this sort of sample characterization service. So you're welcome to contact us if you have a sample that you'd like to have analyzed by AFM, whether it's either a feasibility type study or you just want to have us obtain data for you. We're happy to do that. And again, I just really want to emphasize that we're here to support any of your research needs here, and we can. And this goes all the way from if you just want to independently use the instrumentation to support your own research, or if you want us to help you in the design of the experiment with the procedure and assist in things like data acquisition and data analysis, we're certainly happy to help you do that. So I'd like to thank you all for your kind attention, and hopefully I was able to give you an overview of the type of work we're doing here in our AFM facility. This is my contact information. Please, if you are curious about AFM, if you have questions about whether maybe AFM is the right technique to help you answer some of your research questions, please contact me. Happy to help and coordinate, and even try something, right? And so, again, really happy to help any of you all with any of your research questions or your research needs. And so from here, I'm happy to answer any questions. I do have some pre-submitted questions, which I think maybe I'll start with those just in case if there's some overlap. And so one question was, could you perform AFM of supported lipid membranes on liquid environment? So the answer would be yes, we have the capability to do that. We have a closed fluid cell that we could ramp up to 80 degrees C. So certainly we can support looking at soft materials in a liquid environment. Let's see, I had a few questions that fell into the category of resolution, very specific to material systems. So I guess I'll share that the resolution of these AFMs, so in the Z direction, the resolution is about 0.1 nanometer. And then the lateral or XY resolution, I guess theoretically is 1 nanometer, 2 nanometers. However, I think lateral resolution is really limited by probe geometry, right? And overcoming any sort of like the artifacts associated with from the tip, right, in the tip geometry. And so I don't know, I guess it seemed like some of the questions had to do with some people were interested in antibodies. It looks like some protein analysis here. I guess generally speaking, a large protein, maybe like 500 kilodalton, and with a very sharp probe. And I think also with some further processing, some deconvolution processing, I think you probably could achieve this sort of single nanometer resolution. But I guess maybe seeing is believing, right? So I'm certainly happy to help you all. We could try something. If you have some material you want to submit, we can work that way, and I'm happy to coordinate that further and explore what the resolution we can achieve with your particular material system may be. So that's it for pre-submitted questions. I see some questions maybe coming in the chat. I have seen papers with AFM tips being used as a pH probe. Is that a technique anyone has worked on at UMass? I'm not so sure if anyone has worked with this at UMass. Maybe someone here in this meeting would know better than I would, but I'm not aware of anybody doing this. But I'm happy to explore if you're interested in pursuing this. And so you're welcome to contact me and we can coordinate further. So that was it for the pre-submitted questions. Do any of you folks have any questions that you all would like to ask?
Keith, we have one in the chat.
Oh, sure. Thank you.
In using AFM to measure cell wall from plants' elasticity, I know others have done this elsewhere. It's from Michelle.
Sure, Michelle. Others have done this elsewhere. I guess I'm a little confused with the question, but this is something that has been done here. Okay. And I don't know if the
Michelle, maybe just unmute yourself and ask Keith directly.
Yeah, I mean, I pretty much wrote what was in there. People have been doing this for a while in plant cells, and other than reading papers, I didn't pay attention a lot to the details, but I just don't know if that was something we were set up to be able to do or not, or if this sounded something familiar. Or
Sure, as far as familiar, I guess sort of mechanical measurement of the cell wall, I guess it kind of sounds familiar to me, but I'm not sure if anyone is doing this here. As far as the probes being specialized, I imagine, with any sort of mechanical property characterization with AFM, I think finding not so much the probe, but also just the cantilever sort of geometry, right? Because you can imagine the general sort of stiffness of the cantilever really determines I guess, the probing forces that are available. So,
okay.
Can I chime in here?
Oh, of course. Yes, Alex.
So this type of experiments, they're very they require a lot of time.
Yeah.
And what they require is sample preparation.
Right.
It's sometimes more important than the measurement itself. So I did a lot of cell surface measurements while I was at Purdue. That is 6-month projects.
Yeah.
Not only, so I just want to give the people a little bit of a
Yeah, no, I realize how involved it is, and I'm trying to figure out if it's a path I want to go down, and if, Yeah.
So this is not really something you can give to somebody and then just do it. This is something where a grad student has to be involved. And it's
Right. No, of course, that would be the plan. Yeah.
Yeah. So, but
Yeah.
this is not something which is impossible, but it's just not just making a topography image.
Yeah. No. Right. Exactly. Okay. So it sounds like no one here has done it though before. This is sort of my question: was, has anyone had experience doing it here, or should I go find a collaborator?
The point is, it's the doing is not the point. The point is the microscope can probably do it. Like I said, I did that work at Purdue. It was over 10 years ago. So I did fluid cell imaging.
Yeah.
But
Okay.
I kind of got my head a little bit out of the AFM business, so I got full-time back into electron microscopy.
Yeah.
So
Okay.
I got Keith here. So to
Okay.
fit. But
Thank you.
I know what to do. We can talk about it offline if you want to.
Yeah,
Sure,
maybe.
yeah.
It's sort of one of those things where I'm thinking about, like, should I include this in an upcoming grant, and should I be approaching collaborators, or should we try to figure it out here, or what the deal was? And so I was trying to feel out the experience here.
Yeah, it depends.
Yeah. Okay.
Great, thank you, Michelle. I see another question in the chat here. Does the user need to bring or buy an AFM probe? Yes, and yes, either one, right? The user could bring their own probe, and the AFM core facility, we have a small selection of probes that we can available for purchase. So we have contact probes, good for contact mode imaging, and then we have two different probes for AC or tapping mode imaging. All right, I'm guessing there may not be any questions. But if there are any more questions, again, I'm happy to answer them now. Also, you're welcome to send me a message or contact me via email, and I'm happy to discuss further.
Sounds good. Well, if there's no more questions, then I guess I'll close out our seminar and everybody gets an extra half hour of their day. So I want to thank everybody for attending. Keith, very well done. Lots of information. I hope everybody will contact him to work in his facility. Our next seminar is scheduled for Tuesday, April 20th. We'll be featuring the Center for Human Health and Performance with Director Mike Dusoe. CH2P provides world-class resources to facilitate basic and translational research in physiological, biomechanical, and behavioral mechanisms of human health and performance. So save the date and we hope to see you there. Goodbye, everybody. Thanks for coming.
Great. Thank you.